Hi, I'm Anthony Pimentel.
I'm Steven.
This is the Photosynthesis Lab.
Okay, so you need to get 5 minutes.
Okay, so you're gonna
Here we have a GLX, which is monitoring the level of dissolved oxygen in the water tank. It's connected to a USB cord, which is connected to the laptop. And on the laptop, we're using Data Studio to keep our records. And over here, we have the water tank. Inside of it, there's the magnetic stir bar. And the purpose of this lab is to measure the oxygen output of photosynthesis in aquatic plants.
basically what's happening in the experiment, at least for general photosynthesis, is that the water and the carbon dioxide from the air is being combined with the sunlight in the environment to form oxygen and glucose. In order for these three factors for photosynthesis, you have to have sunlight, carbon dioxide, and water. If not, one of these will not produce glucose. And there are also two different reactions that are present in carbon. There's one in the light and there's one in the dark, called the Calvin cycle.
Go. All right. So right now we have the lamp off on our experiment. So we predict that the plant will just absorb the oxygen in the water, and it will only produce glucose instead of oxygen. But as soon as we turn the light on, we think that sunlight will be added to the reaction and more oxygen will be produced, so our oxygen level will go up. Okay, this is our graph of oxygen level in the water over time. And this section is when we had nothing in the water, so our oxygen probe was only measuring the level of oxygen in pure water. Right around here is where we added the plant into the water. So as we predicted, the plant began to absorb the oxygen in the water. So the oxygen is going down in the actual water sample. And then right over here is where we turn on the lamp, and contrary to what we predicted, the oxygen level is still going down, and then it appears to level out right around here.
Okay, so in here, this experiment, we saw opposite of what we thought. Mainly it's because even though we have the simulation of the sunlight, it's not exactly what the UV rays and what the benefits of sunlight is in the real world. Also, we have a limiting factor of the CO2. Here it's closed off from the outside environment, and the CO2 is needed in order to produce more O2. Instead, just glucose is produced and the oxygen levels drop down. So instead, we would like an experiment either in the outdoors where there's more levels of CO2, or that some way we can pump in CO2 to keep an adequate level of reactants available to the water so that we can make oxygen.
In conclusion, our predictions didn't come out the way we wanted them to, and that's because we underestimated the amount of carbon dioxide that was in the environment, and therefore, since the plant wasn't getting enough carbon dioxide that it needed, there's going to be less levels of oxygen, and that's why in our data, the oxygen levels were just lower and lower.
What's up, guys? It's Jared Jones, doing work over here at Lab Station 1. We're going to tell you how we're going to do some photosynthesis here with a plant in the water. Now, photosynthesis is the action of a plant taking sunlight, CO2, and water and creating sugar, a type of sugar, and O2. And we breathe O2, so this is a very, very, very important process within life on Earth. Tell them, Jared.
You just pass it on to me in the middle of that entire explanation.
Tell him, Jared.
I wasn't ready for anything you were just saying. Frankly,
tell him, Jared.
I'm
Julio Buendia, by the way.
Alright, just redo it.
Keep it.
Keep it.
Let's doing this. Doing this.
No. Hey guys, Jared Jones doing work here at Lab Station 1.
I'm over here with Jared Palmquist.
I'm Julio Buendia.
We're going to tell you a little bit about photosynthesis.
Oh my god, I'm going to kill you.
Oh my god.
What up, kids? It's Jared Jones sitting here at Lab Station 1 doing work.
I'm Jared Palmquist.
I'm Julio Buendia.
And that's it. How you doing, kids? I know a lot of you might be thinking you know a little something about photosynthesis. What we're about to do is a little project on what really happens between photosynthesis. Here we have a project set up. We got ourselves a computer. We got ourselves a GLX. We got ourselves some plants and some water. And a lamp. We're going to be using all 4 of these instruments to monitor how many effects photosynthesis can have on the amount of oxygen within water.
Alright, so as Jared was saying over here, we have a little plant in there, Elodia, and we have the plant in the middle with the distilled water with a stopper over it to prevent any of the O2 from escaping the experiment, so we can actually get a valid and accurate response. And we have the O2 sensor on top just to monitor any change in the O2 that happens actually in the canister.
So basically, photosynthesis is when the sun provides the living energy for plants, which is a complex process involving numerous separate reactions. It can simply be represented by this chemical equation right here. And
Tell them about Are we going?
yeah, we're going.
Hey Jared, tell these little kids about photosynthesis.
Alright, so photosynthesis actually occurs within plants, and it's a function of them actually taking carbon dioxide and water and sunlight in order to create glucose and O2.
So we predicted that with the light turned on, there'll be less oxygen in the container because of the water evaporating, and there'd be a lot more oxygen without the light because of the plant taking in the oxygen from the water and creating more.
Okay.
Okay, so we, as a result, we got that the oxygen went down both with the light on and off. With the light on, the oxygen went down a lot faster than with the light off, and you can see on our graph that this is true.
It did not evaporate.
I'm filming.
Oh, we are?
A light like that did not
Contrary to our predictions, most of the oxygen went down. Well, not most of it, but some of it did. Contrary to our predictions, some of the O2 concentration went down.
Wait, some of it. It's all of it.
All of it went down.
We do that.
Contrary to our predictions, the concentration of O2 went down. We think that it is because there was so much sunlight that the plant was producing O2 at a higher rate, and it didn't have enough time to be fully absorbed into the water, so it escaped into the atmosphere around it.
And we also noticed that as the experiment continued, the plant actually made the water a lot cleaner, and we believe this is due to the plant's ability to absorb the pollutants into its body.
So another experiment we could do to get better results would be maybe to use different kinds of plants, or maybe a bigger container so that we could see the results more clearly. Maybe have different types of probes, because this probe might have had some issues with it.
Hey, I'm Aloysius David Innocentus Macalinau.
I'm Nick Mortal.
And I'm Kenneth Kong, or Pro Darijaya.
Yeah. The purpose of this lab is to find the factors that affect the rate of oxygen reduction in photosynthesis.
So here's our graph, and it shows the dissolved oxygen levels. And here's our equipment. We have one lamp to mirror the sun. We have a stirring bar, something that monitors the dissolved oxygen, and it also contains a jar of water with a smaller jar inside to hold the plant that we're going to use. Okay, so now we just added our plant, which is called Elodia. It's an aquatic plant, so obviously it can live in the water. And we also added the lamp to mirror the sun, so it can act as the photosynthesis for the project.
So, photosynthesis is pretty much H2O, otherwise known as water, going into the plant in addition to carbon dioxide. Now, it needs sunlight to stimulate the photosynthesis, and in turn, it produces sugar and oxygen.
I mean, when? What? When? My prediction for this experiment is that the dissolved oxygen level with the plant will rise as time goes on. That is terrible.
And I think the dissolved oxygen should go up too, because plants make oxygen. Unless it's plastic, then it won't make anything. It'll probably go down.
My prediction is the same, and I illustrated it through this graph. The red line represents the water without the plant. Blue line represents it with the plant.
Looks horrible.
Okay, so here are the results for our experiment. It is against my expectations. This is my graph before, with the blue line going up and the red line staying the same. However, in the actual results, we can see that the red line is actually higher than the blue line. And just as a reminder, the red line is the water without the plant, the blue line is the water with the plant in it.
Okay, so shown in our previous graph, the dissolved oxygen went down, but we do believe that the plant is undergoing photosynthesis. It just creates oxygen that isn't dissolved into the water. If I zoomed in close enough, you could see air bubbles, and there is oxygen being created. It's just not dissolved inside the water.
Experiment better, what we could do is use the plant and seal it off in a carbon dioxide filled room, and use a sensor to measure exactly how much of each gas there is. And so if it does produce any type of oxygen, then the instrument will be able to pick it up.
Okay, and then watch me. Okay. Hey, whoa, I want to do it. Okay. Or you could just, instead of monitoring the dissolved oxygen levels, you could just monitor the oxygen levels as a whole inside the sealed container, because I'm sure it's making oxygen, it's just not dissolved.
So in my opinion, this experiment is pretty much like trying to measure the humidity with a thermometer. It just doesn't work because that equipment is designed to measure dissolved oxygen in water. However, when the plant produces oxygen, it doesn't mean that it dissolves in water automatically. So we need some other type of equipment in order to measure how much oxygen it creates.
So, if we had better equipment or different equipment that actually works for what we're doing, maybe it would have a better result. Do we know? No, because we don't have enough time for that kind of stuff. So, yeah.